Welcome to our third and final video for Chapter 13. In this video, we're going to look at abnormal chromosomes, how we get an abnormal. So we've seen karyotypes a few times before in our course. Remember, a karyotype is going to show us the number and appearance of our chromosomes and includes their length, their banding patterns. You can see different bands here based on the staining that's used. And we can even see the centromere position, especially in the larger chromosomes, it's easier to see. To get a person's karyotype, what cell biologists or Interestingly, chromosome 21 is actually slightly smaller than chromosome 22. So they're usually arranged by length, but it turns out that researchers discovered this after Down syndrome was named trisomy 21. So not to change the name, they just kept the chromosome arrangement like so. Down syndrome or trisomy 21 is when you have 3 copies of chromosome 21 instead of 2. Another thing to note is for our chromosomes, so what are the things that karyotypes can tell us in terms of potential diseases? So one thing a karyotype could show us is whether or not we have an abnormal number of chromosomes. For example, if an individual had Down syndrome, they would have trisomy 21 or 3 copies of chromosome 21, and we would see a 3rd chromosome here. We could also see, for example, if someone has an abnormal number of chromosomes in the sex chromosomes. There's something called Turner syndrome, where a female might have only one X chromosome and the other one would be missing. There would be nothing right here. We can also look at the karyotype to identify large deletions in a chromosome or insertions. So you might see something like this chromosome is abnormally short or abnormally long if there's an insertion. In some cases, you can even identify translocations. Translocations are where chromosomes accidentally swap parts with one another when they're not supposed to. So, for example, a fragment of this chromosome might swap places with that chromosome, and they're not supposed to do that in translocations. So how do we get a weird number of chromosomes or an abnormal number of chromosomes? This arises from something known as nondisjunction. So nondisjunction can happen at one of 2 steps during meiosis. They can either happen during meiosis 1 or meiosis 2. If it occurs in meiosis 1, what happens is homologous chromosomes are not separating as they should, and you can see that here. This is in meiosis 1. If nondisjunction happens in meiosis 2, sister chromatids are not separating as they should. So that's meiosis 2. Either of these cases will result in an abnormal number of chromosomes. So if I look at meiosis 1, this is what happens if nondisjunction happens in meiosis 1. I can see the homologous chromosomes here did not separate. So one cell, the cell on the left, has fewer chromosomes than we should have. Whereas the cell on the right has an extra set of chromosomes that we should not have. If you have nondisjunction happening in meiosis 2 on the right side of the picture, you can see These cells on the left are going to be normal. They have the correct number of chromosomes. And on the right, I see this cell has one less chromosome than it should have, and this one will have one more chromosome than it should have. So do take a look at the number of chromosomes that each of these cells will have if non-disjunction happens in meiosis 1 compared to meiosis 2. Our textbook asks us a nice practice question related to non-disjunction. So they ask which one is true. A, non-disjunction only results in gametes with n+1 or n-1 chromosomes, meaning they're all abnormal. That's not true because I can see if it happens in meiosis 2, then 2 of the cells will be normal, so it's not A. Non-disjunction occurring during meiosis 2 results in 50% normal gametes, which is true, we just said that. Okay, let's look at 3 and 4 anyway, or C and D anyways. Non-disjunction during meiosis 1 results in 50% normal. So none of these are normal, so that's not true. It always results in 4 different kinds of gametes. Not necessarily. See, these 2 are the same, and these 2 are the same. These 2 are the same, so that one is also not true. That leaves me B. If non-disjunction occurs, we're going to get an abnormal number of chromosomes, and this is known as aneuploidy. So one example I mentioned earlier was trisomy 21 or Down syndrome. Often people get confused because they think, oh, having more chromosomes can't be bad, right? That means we just have more information. But that can cause disease or cause certain conditions because we have, for example, a greater gene dose. So if you have too much of one specific protein, that can cause damage or different symptoms and signs compared to the normal individual, which could be deadly in some cases. So most of the time, the smaller chromosomes, if you have more of them, it has a greater chance of survival. Then the individuals have a greater chance of survival than aneuploidy in the larger chromosomes, like chromosome 1 or 2, that often results in instant death. If I look at the terms under here, euploid is when cells have a normal number of chromosomes. Monosomy is when you have only one copy of a particular chromosome. Earlier I mentioned Turner syndrome, which is when females only have one X chromosome instead of 2. That's an example of monosomy. Trisomy is like the one I mentioned earlier for chromosome 21 and Down syndrome. You have 3 copies of a particular chromosome. So it is known that non-disjunction, the risk for non-disjunction increases with age, and the focus is generally on maternal age, where physicians may often mention that after the age of 35, there is a greater risk for having a child with Down syndrome or trisomy 21. And the reason for the focus on maternal age is because females are born with all of their eggs, and the eggs are actually paused. They're in the middle of meiosis, and they do not complete meiosis until every month during the menstrual cycle. So you're going to learn more about this if you take Bio 4B or physiology in the future. This is in contrast to males who produce sperm on a daily basis, although there are risk factors with age as well. Another term to note is polyploidy. So we're used to talking about haploid cells, which are gametes, and diploid, like human organisms, but some organisms display or exhibit polyploidy. For example, triploidy or triploidy is when you have 3 sets of chromosomes in your cells. Tetraploidy is when you have 4 sets. We see polyploidy a lot in plants, another concept you're going to learn more about in 4B, but not in animals. And polyploids are going to look more normal in appearance than an abnormal number of chromosomes. So you have 1 fewer in chromosome number 30, for example. So here in our book, we have an example of a polyploid plant, and it has more petals than its diploid counterpart. Something else that's really interesting is for females, since we have 2 X chromosomes, early on during development, when you're not made of all of your cells yet, every cell will inactivate 1 of your X chromosomes, and that gets tightened up, bundled up into something called a bar body. we can see these in cells. They look like a dense dot right there. So the X chromosome that's inactivated in each of these cells is random. So it could have come from mom or it could have come from dad. So in one cell, maybe this one is inactivated. In the other cell, it could be dad's X chromosome that was inactivated. And this was early in development. So once these cells divide through mitosis, all of the same X chromosome as that original cell will continue to be inactivated. So same inactivated X chromosome. The reason females inactivate one of the X chromosomes is to compensate for that double dose. You have two X chromosomes, but you only want one of the chromosomes to express the genes. So while we're on the topic of sex chromosomes, let's look at aneuploidy of sex chromosomes, when you have an abnormal number of sex chromosomes. And again, this arises due to nondisjunction. So these two examples are males. They both have a Y chromosome, so they're considered genetically male. In Klinefelter syndrome, there's an extra X chromosome. So in these individuals, usually you'll see smaller testes, and these individuals are sterile and cannot have children. You also might see supermales, which is when you have an extra Y chromosome. Usually there aren't well-defined signs and symptoms, although some articles say that individuals that are XYY are more aggressive and may have a greater tendency to be physical or be more violent. In terms of females, you can have trisomy of the X chromosome or 3 X chromosomes. This is also with a frequency of about 1 in 1,000 females. Usually these individuals are healthy and you don't see any distinguishing physical features, although there is a greater risk for learning disabilities. If you only have a single X chromosome instead of 2, I mentioned this one earlier, this is known as Turner syndrome. It's less common. It's about 1 in every 2,500 female births. And usually you see that these individuals are sterile, and also this may also lead to learning disabilities as well. This is the only viable monosomy in humans. So the only case where you can have a single chromosome, so one missing chromosome in your 23 pairs and still stay alive. As I mentioned in humans, for human females, one of the X chromosomes in every cell is inactivated, forming that bar body. Although as a side note, there are some cases where genes on the bar body are still expressed, and we'll talk about that more later. But something similar happens in cats. It turns out the gene for coat color is also located on the X chromosome. And during their development, one of those two chromosomes is also inactivated. For cats, you often see this tortoiseshell pattern because if they have two different alleles for coat color, let's say this is like light brown and this is like darker brown. Why did I choose A? Maybe I should have chosen B. That's okay. And this one is inactivated in one of the cells, you'll express that color. Or this one could be inactivated and you'll express that color. That's when you're going to see this tortoiseshell coloring. Males, though, since males only have one X chromosome, they don't inactivate that X chromosome. They only have one, so they'll never exhibit this tortoiseshell color. Besides nondisjunction, there are other disorders that can cause abnormalities or diseases. So one disorder is caused by a structurally altered chromosome, like the one shown here. If you have a specific deletion in chromosome 5, this can lead to Lejeune syndrome or Creutzfeldt syndrome. So if you have a child that's born with this syndrome, they'll be severely intellectually disabled, and they usually have a cat-like cry. And I believe this is one of the most commonly known cases, this child right here. I believe there are some videos showing his cry to help like medical students understand the syndrome. So children with this specific deletion usually do not survive past early childhood. And again, they exhibit this common high-pitched characteristic cry which is where they named this disorder after. Besides nondisjunction and deletion, we can also have rearrangements of chromosomes that lead to certain disorders. So a chromosomal inversion is when you have a 180-degree rotation, and they can be pericentric. Pericentric is when it includes the centromere. So let's look at the normal chromosome. It's in order A through F, with this being the centromere. A pericentric inversion involves the centromere, and you can see C and D have rotated 180 degrees. A paracentric inversion is when the centromere is not involved, and you can see that's happening here. So for pericentric inversions, this can sometimes change the length of a chromosome in terms of the P and Q arms, whereas a paracentric inversion will not change the length of a chromosome. Sometimes when one chromosome goes through one of those inversions, such as the one shown here in blue, instead of going from 4, 5, 6, 7, 8, you can see 7 is here and 5 is there, so that was the inversion. Then when you're going through meiosis and preparing for crossing over by going through synapsis, the normal chromosome has to form an inverted loop so that the nucleotides, remember the A, C, T, G, and the genes are still matching and in the correct order. This is hard to do, so it's much more likely that chromosomes that have undergone some kind of inversion will align incorrectly with their homologous chromosome and result in faulty synapsis and genes that are not correctly aligned when you get the daughter cells. Finally, the last type of chromosome abnormality we'll talk about are reciprocal translocations. So I mentioned earlier, translocations are when non-homologs switch parts with one another. So completely different chromosomes switch parts with one another when they're not supposed to. One of the most well-studied ones are when chromosome number 9 and chromosome 22 swap with one another. So this should not happen, but if this does happen, this is actually associated with a type of cancer, a type of leukemia we call chronic myelogenous leukemia, or CML. And that takes us to the end of our third video for chapter 13. In the next chapter, chapter 14, we're going to be looking